Drop it.
Hello guys, welcome to my channel Science Tunes. Our topic is about cell structure and its function. It is from 8th standard. So, to explain this cell, I want to give some introduction. See, we see millions of things around us in this world. Some are living and some are non-living things. Living things include living organisms like human beings, animals, birds, insects, so on. Whereas non-living things like soil, air, water, the things that cannot move with their own, those are nothing but non-living things. But living things can do some basic functions like respiration, excretion, digestion, growth and movement. Whereas non-living things like these things, they cannot move, they cannot respire, they cannot digest. But some functions they do. They move from one place to another place. For example, vehicle. Vehicle moves from one place to another place. How? With the help of the living organisms. So, this is the difference between the living and the non-living things. But living things and non-living things are having some micro units called cells. Because with the basic unit, cell is this is cell. Cell is nothing but basic fundamental unit of life. Unit of life. Either the living organisms and non-living organisms, they completely depend upon the cell. It plays a very vital role. Understood? So, our great English scientist, is named Robert Hooke. What is the scientist name? Robert Hooke. He is English scientist. He discovered cell. He discovered cell in the year 1665. How he discovered himself? He discovered self microscope. Himself discovered self microscope. That is, what is the name of that self microscope? Crude microscope. What is the name? Tell me. Crude microscope. With the help of this crude microscope, which he self-designed by Robert Hooke, he is an English scientist. He discovered this microscope in the year 1665. He is so curious how the living organisms are made up of. So he want to know about the cell. So he took one cork. Cork means, you know, what is the meaning of the cork? C-O-R-K, cork. That is bark of a tree. Bark of a tree. In Telugu, I will say, for any plant you take beredu, that means stem. So if you Robert Hooke took the bark of the tree and he divided into slices. He divided into that bark into slices. Okay, slices. Slices means pieces. In bread, you will observe the pieces cutted. That is nothing but a slice. You know very well about that one. So he took that slice of a bark of a tree. That is nothing but a cork. He observed under this crude microscope. So under this microscope, what he observed? He observed some compartments. He observed some compartments. How he observed means, see, he like this, the cork piece slice. This is a slice, and he observed some bits like this, and each one is divided into compartments. Understood? So that he compared with the another one, that is nothing but you know children, honeybees. Honeybees shelter name, what, tell me? Honeycomb. Honeybees shelter name honeycomb. What the honeybee do? It sucks nectar from different flowers and the fruits. The juices where it stores in its shelter. So the shelter is nothing but, here see, honeycomb, whenever you say, I am drawing myself the various what I find the structure I am finding here. So the parts are like this, and every part is nicely, they will design that honeybee is very intelligent. It builds its house very soon. This is honeycomb. What this one, tell me? Honeycomb. So these are the, what these are, tell me? Compartments. What these are, tell me? Compartments. So each structure is looking like a room-like structures. In Latin word, that cell is, they will call them as what, tell me? Little rooms. They call them as what, tell me? Little rooms. Okay, understood. So he observed same thing in the bark of the tree. In like that he recognized the structure of the cell. So for these compartments and these little rooms, what he mentioned, he himself named one word for this compartments. What is that name? Is cell. Cell. The word cell is named by whom? Tell me. Named by Robert. Named by Robert Hooke. Understood? That is my little rooms. Little room. Okay. This is about Robert Hooke. He discovered cell and he identified its structure and its functions under his own self-designed microscope. That is the name of that microscope is a crude microscope. So guys, what happened after 150 years? After 150 years, so much development came. So many scientists have involved. They discovered well advanced microscopes, because Robert Hooke only he identified the structure of the cell. That means how the cell it will be under his self-designed microscope. So that time he named that compartment as a cell. He gave the nomenclature for that compartment by Robert Hooke. But after 150 years, so much development came, and all scientists are very curious to know what is there in that cell, what type of structure it is, and what are the functions it is performing. So they want to know. So they invented different types of microscope. So there are two types of microscopes are there. How many types? Two types of microscopes. So what are those two types of microscopes? So one is the compound microscope. Let us know first compound microscope. Later I will discuss with that another microscope, that is the electron microscope. So compound microscope. This compound microscope is also advanced one. Its magnifying power is 100 to 1500 times. It is having magnification. What, tell me? Magnification. Okay, that means it can observe the thing, that is a slice of any stem or any leaf. If they want to observe under this compound microscope, we can see it clearly 100 to 15 times better. We can observe it, what the parts are there inside. Okay. So, but this compound microscope, where it is available, mostly we can observe in our labs, school, laboratories. There we can see. But compared to this microscope, in this microscope, with the help of the slides, slide means glass slides. Glass slides are used to, glass slides are used to observe the stem, or any item which we are going to see. Okay, understood. Slide, that means glass slides are used, and we can adjust the power also. This compound microscopes. You can see in the figure the compound microscope. Okay, this is the important. The compound microscope is available in labs, schools, etc. And its magnification power is 100 to 1500 times. And for this to observe the any stem or anything, the slice, we are using the glass slides. Okay. And coming to the next microscope, that is compound microscope. Later what it is, they tell me? The most advanced, which is having the high magnification power, that is electron microscope. What is this one, tell me? Electron microscope. This is the advanced one. So advanced. And under this electron microscope, we can see it very clearly. Its magnification power is magnification power is around 1 lakh. Okay, 1 lakh to 5 lakhs, I think. And this electron microscope is advanced one. Its magnification power is 1 lakh to 500 times. It is better to observe the cell very clearly. We can see its structure and what are the parts of there inside the cell and how they are going to function very clearly. We can observe with the help of the electron microscope. But in the compound microscope, glass slides are used. But in electron microscope, electron magnets are used. What they are used? Electron magnets. Electron magnets are used. Okay. I think now you have understood how many types of microscopes are there? 2 types. What are those? Tell me. Compound microscope and the electron microscope. Compound microscope is having magnification power is 100 to 1500 times, we can see it is somewhat clear. But most advanced one is the electron microscope. Its magnification power is 1 lakh to 500 times better, and we can see with the help of electron magnets very clearly. Okay, now you know how to see the cell structure very clearly by using the electron magnets. So scientists discovered. Later we are going to discuss about the cell. Okay, this cell is now we are entering the topic about the cell. Before considering the cell, before knowing about the cell, I want to discuss with you our human body. Our human body is made up of a number of organs. To form an organ, first starting is a only single cell. It starts its life with a single cell. So that's what we are calling it as a cell is a fundamental unit of life. Fundamental unit of life. Cell is the fundamental unit of life. Any organ is for forming its own shape means with the help of the cell only it starts. See, I am drawing here diagram. See, this is a cell. Okay, this is what, tell me? Cell. This is single cell. What, tell me? Single cell. Single cell, it multiplies its number. It multiplies its number. That means this is nothing but what, tell me? Zygote. What is this, tell me? Zygote. Zygote is nothing but the fertilized egg cell. Fertilized egg cell. Okay. So single cell is entering that multiplying into two cells, and two cells are multiplying into three more cells. A group of cells together form into tissues. Tissue. This is what, tell me? Tissue. A group of tissues. A group of tissues together form into an organ. What, tell me? Organ. A single cell, any living organism or non-living organism, they start their life with a single cell. Then they multiply into number, they form into a tissue, and a group of tissues together form into an organ. A group of organs together form into a organism. Understood? Organ system into organism. In our human body, many organ systems are there, like digestive system, a respiratory system, excretory system, and reproductive system. Understood? And that means a group of organs are working together to perform a certain function. Okay. Now we can discuss the topic in the next topic. So guys, today I think whatever I explained, I think you have understood. So next part I am going to explain about its structure and its function. What are there inside the cell, I am going to discuss with you. So don't forget to subscribe my channel and ring the bell. Okay, thank you.